In this next session of the Covering Rare Diseases Fellowship Program, we're going to talk about the pathway from rare gene discovery to actual drug development. We'll hear from Dr. Michael Collins. He's a senior investigator, skeletal disorders and the mineral homeostasis section of the NIH's National Institute of Dental and Craniofacial Research. And thank you for joining us, Dr. Collins.
My pleasure. Thank you, Rachel and Sunny, for asking me.
In my first conversation with you, you helped provide me with some very valuable context about things that certainly journalists need to understand when communicating rare disease research. And that is the fact that sometimes progress seems incremental, but other times it can seem explosively fast. But the first question I'd like to ask you before you do your presentation is, do you feel we're at a sort of inflection point in terms of the promise of rare disease research?
Absolutely. You know, I've been in this business now for over 20 years. And for most of that time, I would see patients, I would do my best to take care of them. And the fact is, most of the time I had little to offer them. And that was frustrating for me. That they appreciated just someone taking care of them and someone who understood their disease. But within the last 5 to 10 years, there's just been an explosion in advancement, and I'll talk about how that's come about. But I absolutely think we're at an inflection point. I think there's great promise for patients with rare diseases, and reporting is absolutely essential for that progress to continue, I think.
Great. Well, why don't you share your presentation with us and get us started?
Okay. Get this out of the way. Okay. All right. Once again, thank you, Rachel. Thank you, Sunny, for inviting me to do this. I'm really honored to be here. As I said, I think that reporting plays a really critical role in advancing what I do. So I appreciate all you do. I am taking the presenter's prerogative to slightly change my title from the one that you see here to this one, which I think really captures a little bit better what I want to talk about. And that's how we are, where we are in terms of treatment for patients with rare diseases. I do have a couple of disclosures, which I'll show you here. And then I'll just move on to the way I'm going to approach this today. So I wanted to give a little bit on the rare disease drug landscape. I suspect that probably much of this you've heard or will heard, so I won't spend much time on it. What I wanted to really focus on are the 3 case studies in rare diseases, which I think really reflect the spectrum of how things are going these days. And to do that, I'll have to tell you a little bit about my group at the NIH, the Skeletal Disorders and Mineral Homeostasis section. Okay, so for the landscape. So just simply to get sort of definitions out of the way, again, you may have heard this, that in the US, a disease is considered rare if there's less than 200,000 people. In the EU, it's less than 5 per 10,000. Japan, less than 50,000. So that gives a global average of about 1 in 2,500 patients. The number of rare diseases, depending who you listen to, is a little bit all over the map, somewhere between 5 and 10,000 rare diseases. And probably the best current estimate of prevalence is about 6.2% of people. So in the US, that will be 20 million people. In the US, 47 million people. And to give you a sense of the cost of the drugs for caring for these patients, I've shown you this from the Weeva Institute here, where that shows you the rise and increase in expending in rare diseases, which reflects the fact that we now have orphan drugs to treat patients with rare diseases. And this has really come about from what I consider to be a sea change that's taken place mostly in the last 10 years or so. I'm sure you heard earlier about from Eric Green about the advances in genetic diagnostics, increase in speed, decrease in cost, and increase in accuracy, advances in rare drug discovery. We started a program to look for small molecules to treat fibrous dysplasia, and we had a library of about 200,000 molecules. We thought we were really doing great, and now these libraries are up to trillions of molecules to discover drugs for patients. These are in silico, that is, they're done in computers, which is really much less expensive than those that we had to do in dishes in the past. These molecules eventually need to be tested in preclinical models, primarily mice, and the CRISPR mice advances have made this a much more rapid development than it used to be in the past. There's also been changes in the economic landscape. The entry of small biopharmaceutical companies into this, the interest of big pharmaceutical companies as well, legislative incentives for companies to get involved. And I think there's been huge advancements in patient information and support brought about by changes on the internet, including social media and patient support groups. And all of this has worked together to really, in the last 10 years, as I said, especially to change the whole map of rare disease treatment, much to the benefit of the patients. So the way I wanted to approach this today is I wanted to talk about 3 different diseases and treatments of patients that I see in my group at the NIH. And the first, I'll just outline them here. This is tumor-induced osteomalacia, where we have used a drug called infigratinib. And this was really a very rapid application, a repurposing of this drug that was in development. So this is one sort of model that's happening now. And this is one of the ways in which drugs are coming to treatments for patients with rare diseases. The second is another disorder called autosomal dominant hypocalcemia type 1, where we're using a drug called incalerat. This was more of a protracted application, a repurposing of this drug, of an existing drug, and I'll tell you how that has happened. The one I'll finish with is the McCune-Albright syndrome, fibrous dysplasia, McCune-Albright syndrome. The drugs are yet to be determined. We don't have drugs for this yet, and this has really been a tough nut to crack, and you'll see why. So while there's been great advances, there's much to be done, and there's not a panacea approach to all these. It's quite different depending on the drug, the patients, and the underlying nature of the disease. So I wanted to tell you a little bit about my group at the NIH and why we do what we do and how we got to these diseases that I've outlined here. And so my group is called the Skeletal Disorders and Mineral Homeostasis section. And we study disorders of parathyroid hormone, PTH, and fibroblast growth factor 23, FGF23. Well, why do we study those? Well, my group is interested in mineralized tissue, bones and teeth. And if you take a biopsy of a piece of bone, it looks something like this. And you'll see that on this biopsy, there's very few cells. These little arrows point out the cells. And most of what you see there is what we call matrix, the stuff in between the cells. In this matrix, the hard stuff that makes up bones and teeth is this molecule called hydroxyapatite. Hydroxyapatite is a biological crystal that's made up primarily of calcium and phosphate with a little hydroxyl group in the middle. And so FGF23 and PTH are the hormones that regulate calcium and phosphate in the blood and into bone, and to regulate the formation of cells that make up bone and all the hard tissues, bones and teeth. The way that my group approaches this is shown here. So we see patients with rare diseases, and we've seen a lot of these patients over the years. And one of the main goals in this group is to listen to them and to hear from them and to identify their unmet needs. We do this in natural history studies that we conduct at the clinical center at the NIH. Sometimes these natural history studies lead directly to clinical trials with repurposed drugs. More often, these involve translational studies in the laboratory, where we try to understand the pathophysiology that underlies the disease. We develop models to do preclinical testing to understand and to have models to treat, test molecules in for preclinical testing. And then this is called a sort of bed to bench to bedside approach. So we see the patients, we work in the laboratory, and back to the patients here. And really the goal is to reduce illness and disability, which is part of the mission of the NIH. Now, how long does this take? Well, it depends, but this is probably a fair estimate. Sometimes these natural history studies can go almost directly to clinical trials. More often, it's a lag time of 5 to 10 years of figuring this out in the laboratory, and then another additive 5 to 10 years back to the clinical trial. So you can see this is really a fairly long process most of the time. And I'll show you how this is sometimes expedited with luck, and how more often than not there are barriers that get in the way, some of which we are getting through, and we can get through yet again better in the future. So first one, tumor-induced osteomalacia, and the drug that we've used to treat this condition is called infigratinib. So TIO is a disease of FGF23 excess. FGF23 lowers blood phosphate. So when you have excess FGF23, blood phosphate is quite low, the bones are quite weak, and some of the patients can develop these horrible fractures that you see in this poor guy here. Now, TIO is an interesting disease. It's caused by these usually very small and usually very difficult to find tumors. This white arrow here on this scan of this patient is pointing to a little teeny tumor. And this tumor caused terrible disease in this patient. And just excising this tumor, which in this case was quite simple, cured him. So tumor removal is curative. However, some tumors are very difficult to find, can't be found, and sometimes they escape the normal control and end up with metastatic disease, as is shown in this patient here with these arrows pointing to multiple metastases. And this man had a very profound problem with this disease. Interestingly, and of interest to you guys, is that this patient actually was a journalist. He was a TV journalist. I don't think this is a job-related condition, but an interesting coincidence for today. So, how did we figure out and crack the nut with TIO? Well, in 2014, we participated in a study with a group in which the underlying genetic defect that causes tumor-induced was identified. And this was brought about by advances in sequencing. RNA sequencing of tumors found this defect in 2014. And what this defect is, it's a different kind of mutation called a translocation. So this is where one part of one gene now is broken off and connects to another part of another gene. And in the case of TIO, the one gene was this sort of structural protein called fibronectin 1, and it is now hooked up with this signaling protein called FGFR1, fibroblast growth factor receptor 1. And it was possible, we thought, that this FGFR1 signal is transmitted to the cells in the cause of this condition. And so we figured that possibly an FGFR receptor blocker may be effective. So we were able to start a compassionate use study in a molecule that was available with Novartis at the time that was called BGJ398, that went on to become the drug infigratinib, eventually approved for cholangiocarcinoma in 2021. And it's a sort of pan-FGF receptor inhibitor. So there's a number of FGF receptors, FGFR1, 2, 3, but this molecule blocks all of the FGF receptor inhibitors. And we started treating this patient with this drug in 2015. And as you can see, he had an almost immediate and dramatic response to this drug. All of these dots that you see here, this shows where this went away, and most of these dots went away. And he had a very dramatic response, tremendously improved the quality of his life. We went on to treat this patient for 5 years. And unfortunately, one of the problems with this drug, this is a problem with a number of these drugs, is there's a lot of side effects. So we started him on treatment. We treated him for 1 year. He did a lot better. He got back to playing tennis again. He got to see his daughter graduate from high school. But eventually the side effects were so severe that he just couldn't take it anymore. So we stopped the drug. He was off the medication for 28 months. And as you can see, this disease really just came roaring back. We initiated treatment again and again. He had a very dramatic response. But ultimately, the side effects were too much. He couldn't take it, and he succumbed to his condition. But nonetheless, he was able to get 5 years of, for the most part, fairly decent quality of life out of this. So this was really quite a success story for him and for the field, I think. And so infigratinib represents our experience with infigratinib that I've gone over here represents one way in which we get to treating patients with rare diseases. So if we consider this as a model of a disease treatment, the TIO had been around for greater than 50 years. The genetic underpinning identified in 2014 came about through advanced molecular sequencing with the FGFR1. FGFR inhibitor drugs first appeared in the 2000s, first approved in 2019. And this was another advance that allowed us to latch on to that. So this was a repurposing of an existing drug. We went on to conduct a trial in a group of patients with benign TIO. And then here again, we had to stop the study due to toxicity. But I do think that in the future, we can have more selective drugs that will have less toxicity, less off-target effects, as we talk about. And you've probably heard about, or will hear about more in the future, there are the potential for oligonucleotide therapy in this patient. And what is needed in this and many of these diseases of tumors or mosaic disorders is tumor-directed delivery. And there are ways that is coming about as well. I wanted to move on to the second case study of a disease, and that is one called autosomal dominant hypocalcemia type 1. And I wanted to start out this story with one of the patient's stories. So this is Mr. B's story. So this condition is caused by a germline genetic mutation. So he had this all of his life. As a child, he frequently experienced cramping, paresthesias. He often had foggy thinking. He was falling behind in school, wasn't doing well. Eventually, at the age of 6, he was evaluated for learning difficulties and found to have low blood calcium, and he was diagnosed with hypoparathyroidism. Interestingly, in his case, and this is not uncommon, his family history showed that his father, when he was checked at the time of the diagnosis of his son, also had low blood calcium. So this is where he got it from, and this was the manifestation of the genetic nature of this disease. But what to me was really striking about this man and his family is he had 2 siblings who died in infancy from probably hypocalcemic seizures. So this is a family that really suffered tremendously from this condition. Once he was diagnosed, he went on to treatment with medications that were available, calcium and vitamin D, which can keep the blood calcium up. But one of the problems is that there's really high levels of blood calcium in the urine. And these patients develop nephrocalcinosis, as is shown here on the CT scan of his kidneys, where you can see this white outlines area of his kidneys that have become calcified. This can progress, and some patients can actually lose kidney function from this. And we've actually had patients who've also needed to have kidney transplants. So I wanted to talk a little bit about autosomal ADH1 so as to introduce you to the way that we arrived upon treatment for him. So ADH1 is caused by low levels of parathyroid hormone. Parathyroid hormone is essential for maintaining blood calcium levels, but parathyroid hormone levels are regulated by what's called the calcium sensing receptor, which is on the parathyroid gland. So this is one instance where the medical terms make sense. The parathyroid gland makes parathyroid hormone. Calcium sensing receptor is on the parathyroid gland, and it senses the level of calcium and controls the release of parathyroid hormone. So ADH1 is caused by a variant or a mutation in the calcium sensing receptor. And this was first identified in 1994. And what happens in ADH1 is the calcium sensing receptor is inappropriately turned on, such that it lowers parathyroid hormone levels. Now, the calcium sensing receptor was first discovered in 1993 by two men, Ed Nemeth and Ed Brown. And relatively quickly after that, so this was pretty fast, agonists of the calcium sensing receptors were discovered in 1996. What we need for ADH1, though, is an antagonist to turn that off. And these were reported first discovered around the year 2000. So there's an interesting phenomenon that took place in terms of how this drug got back to ADH1 patients. So one of the properties of this calcilytics is that they represented potentially a way to treat osteoporosis. Common drug, the billion-dollar drug for these patients. So multiple companies developed calcilytics for the purpose of treating osteoporosis. In 2005, GlaxoSmithKline licensed a group of calcilytics from this company called NPS Pharmaceuticals to develop drugs for treating osteoporosis. Now, we knew from the very beginning that calcilytics, these antagonists of the calcium sensing receptor, had the potential to treat ADH1 and had been eager to get these for a long time. So in 2007, we negotiated a contract with GlaxoSmithKline to use their calcilytic ronocaleret to treat patients with ADH1. Unfortunately, 2008 GlaxoSmithKline drug ronocaleret failed, and GSK abandoned ronocaleret, and that was the end of the study that we had started with them. One of the phenomenons in this area is that when a drug company is developing a drug for a disease, and especially if it's developing a drug for a common disease that has a potential big payoff, they're really careful, they're really not eager, they're really afraid of doing other studies with that drug, for example, in rare diseases, because any untoward effect that occurs using that drug in a rare disease will slow down the progress of the study in the common disease, and that has significant financial implications for the company. So this is a barrier that I think can be overcome, but it was a barrier that we encountered here. So 2011, NPS Pharmaceuticals licensed these calcilytics back from GSK, and we negotiated a study with NPS Pharmaceuticals to do this study at ADH1 in 2014. We did an initial proof of concept study that looked very promising. We were ready to move on to a larger study. But at that time, 2015, Shire Pharmaceuticals acquired NPS. They had very little interest in ADH1. Things were getting off the rails. 2019, Takeda acquired Shire. They had no interest in ADH1, so that went bust. In 2020, I approached a small biopharmaceutical company in California called Bridge Bio about the potential to treat patients with calcilytics. Recall, many of the pharmaceutical companies had these calcilytics that had failed in osteoporosis on the shelf. So Bridge Biopharmaceuticals contacted a company, Japan Tobacco Company, that had a calcilytic that they had developed for osteoporosis. It was a really lousy drug for osteoporosis, but it ended up being a great drug for ADH1. And so we've conducted a phase 2 study at the NIH. We started it in 2020 and we finished it in 2022. So that's pretty fast. And you can see the main effect here. So these patients have very low blood calcium levels. These can be very problematic. They can lead to tingling, cramps, seizures, and if you're hypocalcemic long enough, you can actually have respiratory depression that can be fatal. Anyway, this drug proved very effective in this phase 2 trial. And a phase 3 study, multi-center study, is going to be started at the end of this year or the beginning of next year. So for ADH1, again, the calcium sensing receptor gene and the ADH1 variant were identified in 1993. So not too long before drugs were developed, the year 2000. However, the phase 2 study was really delayed until 2020, so almost 20 years. And this was because of the inability for the pharmaceutical companies to take risks on off-label use of drugs in development. And in the case of as I showed you, NPS and Shire and Takeda, acquisitions, mergers, and lack of interest and competing interests for drugs. It ended up that Takeda and Shire also had another drug that was available to treat hypoparathyroidism, which this would have represented a sort of internal competition for that same group of patients. But the phase 2 study was enabled by the new phenomenon of small pharma interest in rare diseases, FDA incentives, and the fact that we had a well-studied rare disease cohort at the NIH. I'm going to move to now, I think we'll have plenty of time for questions at the end. The final case study, which is fibrous dysplasia McCune-Albright syndrome. Now, this is different than probably most of the genetic diseases you've acquired. This is a mosaic disease, and these are somatic mutations. So none of these patients get this variant, this mutation from their parents. These variants, these mutations arrive somewhere early in development, shown here in the blastocyst stage. And then as this mutation occurs in a single cell, and that cell divides and separates out as the organism begins to form in what we call the gastrullophage and eventually the embryo. And so this map of affected tissues in these mosaic disorders is charted in utero. And the affected tissues that a patient have are determined by when and where these mutations occur. These mosaic diseases are the more rare of the rare diseases, they're the more challenging of the rare diseases, and they're some of the most complex of the rare diseases. And you get a sense of that shown here. This is a sort of a panel of a number of problems that these patients can encounter. A lot of hormones are regulated by this gene, and you can get too much of the hormone and too much growth or too little growth. You can get these pigmentations on the skin that reflect the fact that this is a mosaic disease. And this is where this concept of mosaic comes from, like a mosaic painting. In this case, the thyroid is involved, as you can see here. But the most vexing problem for these patients is the skeletal disease. They get these sort of tumor-like lesions in the bone that can be very painful. They're prone to bending, prone to fractures. It's a really very challenging and difficult disease. So the cause of this, and this is part of the problem, it's a really difficult to target protein. So it's this protein that's called GS-alpha, and it's shown here. And this GS-alpha is a major regulator of what we call G protein coupled receptors, which are really important in all kinds of biology. There's more than 800 GPCRs in the body, many of them coupled to this GS-alpha. And then from that, there's downstream signaling from that, which again affects many tissues. In the endocrine organs, there can be excess hormone. In the skin, excess melanin, the pigmentation that you see. And in bone, you can get this condition, this overgrowth called fibrous dysplasia, as well as excess FGF23, which tends to make the bone soft and prone to fracture as well. So how are we going to treat this? Well, for a number of complicated reasons, the molecular targeting of the mutation in GS-alpha has proven very difficult. The disease was first described in the 1930s. It took 50 years before the GS mutations were discovered in 1989. Again, that's part of that is because this is a mosaic disease. So many diseases, you just take a swab from the mouth, you take white blood cells, you can make the diagnosis. In this condition, it wasn't apparent for some time that you actually had to get the affected tissue. So in these patients, most of the cells in their body are completely normal. You have to look in the affected tissues to find the mutation. And so this mutation, as the cause of FDMS, wasn't discovered until 1991. Here again, because you have mutant cells and you have normal cells and you have affected and unaffected tissue, the drugs have to be very specific. You have to target only the mutant GS-alpha to avoid the off-target effects. Then, unlike many of the mutations where the mutated protein is on the cell surface, this is inside the cell. So you need to get this inhibitor inside the cell. And we worked on this for a long time. We had a very active collaboration between my group and the NCATs. I think you're going to hear from some of the NCATs later. And we really beat our head against the wall for this for almost 10 years. We had an active collaboration from Brian Kabilka at Stanford, who was a Nobel laureate working on G proteins. And it was a very frustrating problem that we eventually had to give up on. Fortunately, and this is published this month, October 22, 2022, by Kevan Shokat at UCSF, he has just recently discovered peptides that target this mutant GS. So this is the GS here. It's got its normal protein here, the GS-alpha. And these cyclic peptides are little round, flat molecules that when you flip on their side, fit right into this little cleft right here. And this is part of why this has been difficult to target. This cleft is usually closed. It opens and closes, and a molecule has to fit perfectly in there for the little bit of time that it's open. And it really looks like Kevan Shokat has developed molecules that really hold great promise for targeting this disease. Now, he's clearly shown that he has molecules that can treat this wild type GS-alpha and this mutant wild type mutant GS-alpha also, but there's yet to be demonstrated mutant specificity. So there's still work to be done, but there's good hope. Okay, so FD-MAS is a rare disease treatment model. Again, the GS-alpha gene variant was identified in around 1990. These mosaic disorders are a lot harder. G protein associated mutants have been very difficult to target. And 30 years in now, we're only at the stage where we finally have promising molecules. However, treatment studies will be facilitated by the fact that we do have what are called drug-ready cohorts. So we have a detailed long-term patient natural history study of patients with FD-MAS, FDNH. We have a patient registry, and we have validated in vitro and in vivo models in which we can test these molecules for treatments. So, rare disease treatment is challenging, but the barriers are falling. The genetic causes are identified much sooner. However, there's a lot of time between discovering the gene and coming up with a therapy. The drug screening libraries are much larger than they used to be. Nucleotide and gene therapy approaches are improving a lot, and you're hearing about those later as well. There are legislative financial incentives and small pharma's interests as well. We still need funding for natural history studies and international collaboration. These are expensive. Creative trial design and FDA agency flexibility to accommodate the small numbers of patients and the lack of traditional control groups. But what's also helping is what you guys are doing. Great reporting to inform the public, to support the patients, and sway the funders and legislators will do a lot towards moving us to treatments for rare diseases. And with that, I'll close and take any questions that you have.
We do have a question in the channel, and it's coming from one of our fellows, Hawken Miller. Hawken, would you like to ask your question?
Yes, thank you so much, Dr. Collins, for your time today. I am a fellow and work on rare diseases and writing about rare diseases. And one of my focuses is talking about data and using data and analysis to discover new treatments. And you kind of talked about drug repurposing. But I was curious to hear more about how drug discovery in silico works. That's actually the first time I've kind of heard that terminology. You hear in vivo, you hear in vitro a lot, but that was interesting to me. And I'm curious how that works and how that accelerates the discovery of new rare disease treatments.
Yeah, that's a good question. And it is a relatively modern phenomenon. In silico is kind of a, a play on words. In vitro is in a dish, in vivo is in an animal, and in silico is in silicon. So it takes place on a computer. It's a virtual search. And so what's happened is and this is big data. So the structure of these molecules has been, that's also made tremendous advances as well. We used to only be able to get crystal structures from crystallizing the protein. There's new ways now with advanced microscopy where we can get the structures of these molecules. And so these structures can be modeled on computers, and then virtual libraries of molecules, from small molecules to large molecules to cyclic peptides, can then be, through sort of kind of trial and error, tested for how they'll fit where you want them to fit to bring about the changes that you want in the molecule. Now, these small molecule and silico libraries are literally trillions of molecules. So, and in fact, that paper that I cited there, that Cell paper, where Shokot was the last author, he talks about the process in this paper there, and the approach that they took to from the structure of the protein that they knew to the molecules in this library that they developed, to sort of virtual trial and error, find the molecules that fit in the place that you want them to fit. So that's in silico modeling. Now, still, you find a molecule that you think that works. You still have to do in vitro testing with cells. You still have to do in vivo testing with animal models of these molecules, and you still have to do clinical trials. So that part is still pretty slow, but on the front end, this sort of discovery within silico testing has really advanced us to getting to molecules that have promise. Does that help answer your question, Hawken?
Yeah, absolutely. And, maybe something to talk to you later about, but, the use of artificial intelligence in that as well is interesting.
Absolutely. It's having a tremendous impact on the field.
Michael, I have a question based on some of our earlier sessions today, and many of our speakers acknowledge this sort of gap in knowledge between Western developed countries and lower middle income countries, particularly when it comes to data and information about prevalence, et cetera. So if we were to factor in that issue when it comes to the rapid advancement or the promise that we have, how do we characterize that promise and that advancement when we still have a long way to go in terms of parity and access to data?
Yeah, no, that's a great question, and it's a huge problem, and it's just not right. I mean, you saw when I gave the estimates for the prevalence of rare diseases, what do we have good data on? We have good data on the US, the EU, and Japan. Come on, where's the rest of the world? For the individual, I would say things have opened up a bit. Not a week goes by when I or my colleagues aren't contacted from someone from China or Africa or the Middle East, whose resourceful parents get on the internet. They, through open databases, find out where the people doing the research are. They figure out how to get our email addresses, and they email us and they ask us about their kids. So, we are able to provide some information, but, the fact is, information helps a lot, but treatment for many of these things still is not available where they are. Much less do we know the natural history of the prevalence, and much less do we have centers of excellence for patients to be treated in these countries. So, I mean, it's really heartbreaking. I wish I knew the solution to it, but you pointed to what's a huge problem. Again, I think this is a way where information and reporting is a huge part of getting information out there, can help to move the needle on this, but it's a big problem.
Well, in that vein, even in Western countries, sometimes the cost of drug can be prohibitive. And so how do we address that issue? Are there any efforts underfoot to try to deal with that as well?
Well, you've touched on another huge problem. The cost of some of the drugs for these rare diseases is astronomical. A number of my trainees come from places from South America, other places like that, where some of these wonderful drugs that are now available for rare diseases just aren't available through the public healthcare systems. And it's heartbreaking. They're not available because they're prohibitively expensive, prohibitive to those healthcare systems. How do we get those costs down, Rachel? I'm just a doctor. I don't know how to. I mean, that's a huge problem. Come on, these are real challenges that we really will take commitment to make them change as well. But you're right. This is an unfair practice, if you will, that exists today.
I have a question here. It says, are there animal models for MAS? And if so, has gene editing of the mutated gene been attempted with CRISPR technology?
That's a great question. And there does exist an animal model for FDMAS. And this is one of the big divides in thinking about rare diseases. Keep in mind that FDMAS, like a number of rare diseases, is a mosaic disease. So that means that a lot of cells in the body aren't affected. So theoretically, a CRISPR edit will only target the mutated gene. But in fact, many of our edits and many of our drugs as well also have some degree of off-target effects on the wild type. So mosaic diseases offer a particular challenge in this way. That said, gene editing does represent a viable treatment. Oligonucleotide therapy represents a viable treatment for patients with FDMAS as well. It's not been a tack that we've taken simply because we don't have the resources to do much beyond what we're doing at this point. But yes, CRISPR editing does have the potential to treat a disease like FDMAS. And when the technology gets better, and when we're able to more effectively target genes, especially without any untoward side effects, then benign conditions such as FDMAS will certainly become a reasonable target.
Hello.
Hello, Namat.
Namat Kradli from Lebanon. Thank you very much for this valid information. Well, do you agree that authority determines knowledge? Like, for example, in the Middle East and developing countries, some rare diseases are not taken into consideration because people in power, like, for example, universities in the Middle East, especially official universities, are dominated by politicians, people in power, and they determine what rare diseases need to be treated. So do you agree that this is true? Some rare diseases have priorities in order to be treated. Thank you.
Well, yeah, I mean, it's a good question. And, I don't really know the answer. I suspect that it is the case. I've treated a number of patients from the Middle East to a young girl from Palestine. And it was really, really hard. There were a lot of barriers to getting her out of Palestine through Israel to the US to be able to offer her treatment. It was very difficult. And I suspect that this arises from, at least in part, from the powers that be ineffectively regulating the way business is done. I think that's probably true.
We are out of time, unfortunately, but there are other questions that what we'd like to ask you Dr. Collins, is if we forward these questions to you, perhaps we could keep the conversation going and get the answers to the journalists before the end of the fellowship.
I'd be glad to do that. Feel free to send them.
But at this point, I'd like to thank you for providing us with much needed context when it comes to covering the treatment and the possibility of reaching an effective cure for people with rare diseases. So thank you for making time to join us today.
Thank you for having me, Rachel. I appreciate it. Take care. Bye-bye.
Take care.